very warm welcome to the lab house, everyone. Today let's talk about serology. What is serology? It is the branch of immunology that deals with the study of serum, especially focusing on antibodies and their interactions with antigens. So these antigen and antibody interactions are useful in the diagnosis of infectious diseases like HIV, typhoid, hepatitis, etc., or in blood typing and transfusion safety in order to prevent mismatched blood transfusion or transfusion reactions or transplant rejections. We will perform serological test or in autoimmune diseases. Most importantly, for example, ANA test, that is basically for the autoimmune disease named SLE, systemic lupus erythematosus. Serological test is needed for its diagnosis, or hormone level detection, for example beta hCG hormone that is produced during pregnancy in large amount, or vaccine evaluation. As we all know that in vaccine we will introduce a weak antigen in our body. So in order to generate or detect an antibody response against that weak antigen, we have to evaluate the antibody titers. So this is vaccine evaluation and it also includes serological tests. Now, what are the several ways or several kinds in which these antigen and antibody interactions takes place? So these include agglutination test, precipitation or immunodiffusion test, ELISA enzyme-linked immunosorbent assay, Western blotting or rapid diagnostic test. For example, for dengue, malaria, kits are used for the rapid diagnostic test. We will discuss all these tests in the later videos. So how the immunity is generated? Immunity is generated due to the entry of antigen, but the antigen should be an immunogen. We will discuss the difference between antigen and immunogen in the next video. So the antigen, which is an immunogen, can generate the immune response that is discussed below. So the immunogen entered the body. It is presented on the APC, antigen presenting cell, that can be macrophage, dendritic cell or B cell. So this antigen presenting cell will further activate the helper T cell. The helper T cell will further help in the activation of cytotoxic T cell and B cells. Cytotoxic T cells will do killing and B cells will produce antibodies. Let's understand this with the help of a diagram. As you can see, this is a bacteria and the macrophage will engulf it, phagocytose it and present it on the MHC2. The antigen is presented in the form of peptide on MHC2. So this macrophage will become an antigen presenting cell. This antigen presenting cell will release interleukins. Interleukins are cytokines, those proteins that help communicate the immune cells for their activation. So interleukin 1 is released that will attract the helper T cell. The helper T cell gets activated, as you can see, by communicating with the APC. So the activated helper T cell will further release interleukins. For example, interleukin 2 for the activation of further helper T cells and cytotoxic T cells, or on the other hand, interleukin 4 and 5 for the activation of B lymphocyte, which will eventually come plasma cell, and the plasma cell will produce antibodies. So this picture sums up the whole immune response that is generated when an antigen or immunogen entered our body.